Last but certainly not least, Dr. Arnaud de Lobel is our third speaker, and he is currently the R&D director at Quality Assistance. He oversees internal R&D projects aiming to implement new analytical technology that ultimately brings benefits to the customers of Quality Assistance and in return further enhance the abilities of Quality Assistance to develop new R&D services. His presentation today will focus on how DryLab and Empower function as tools to implement QBD principles to optimize methods for large molecule subunit analysis. Arnaud, welcome.
So thank you, Tran, for this very kind introduction. So today I'm talking about DryLab and Empower and the way to combine both to optimize the analysis of map subunits. So I'm Arnaud de Lobel, I'm R&D Director at Quality Assistance. And very few words about Quality Assistance. So we are a service provider, contract research organization, providing analytical services to the pharma and biopharma companies. We are located in the south of Belgium, and all our labs are located on one site, so here in Belgium. And we provide services for all kinds of products, from small molecules to monoclonal antibodies, antibody drug conjugates, cell and gene therapy. And all the work we do is done in a GMP or GLP environment. So if you have any question, if you want to know more, you can go on our website and you will find all the information you need. So first of all, I will explain what is DryLab and how it can be used for HPLC method development. So there are currently different approaches if you want to develop an LC method. So the first one is trial and error. So you will study one factor at a time, and you need repeated and varied attempts that will continue until you obtain a method that is compatible with your expectations. It can take time, and usually you don't obtain the optimum method at the end. The other possibility is to use experimental design, also called factorial design. And in that case, you have a black box and you have an unknown relation between your input variables and the response you obtain on your chromatogram. Another possibility is to base your development on structural considerations, and then you will base the development on log P, log D, pKa of your sample, of your molecules. And then once you have all this information, the retention can be predicted for a given phase system. And finally, the last approach is retention modeling. So you will use the behavior of the common solutes that is well known. So you know that for proteins, they will behave some way, oligonucleotides will behave another way. And there are well-established models that are available if you want to use this kind of retention modeling. And that is the approach that is used by DryLab that I will present here in this talk. So why would you want to use retention modeling? So first, you do not need to know the structure and the properties of the molecules to be separated. Sometimes you have some impurities for which you don't know exactly the structure. And anyway, you have to separate them and develop your method to separate this molecule from another one. The prediction is based on well-established empirical models. And you will only need a few initial experiments, so usually between 2 and 12, to be performed. And once you have performed these initial experiments, you can predict the separation in any conditions. And so the changes in conditions can be studied in silico. So for example, a change of column length, a change of flow rate, a change of gradient time, you will be able to predict the chromatogram you obtain if you change those parameters without having to do any additional experiment. So here is the workflow that is used with DryLab. So everything that is given in blue can be performed into the software. So first, you will have to do outside of DryLab some preliminary experiments in order to fit into the linear gradient range. Then, once you have that, you will perform your design of experiments by selecting the method parameters. Then you will run the input experiment. So as I said, between 2 and 12 basic input runs. Then you will model, you will optimize your model in the design space. So you will, in that step, change the conditions in order to get in silico the best separation possible. And then you will select your set point, so the conditions that you think are the most suitable, And then you will verify these conditions experimentally to check that the modeling was correct. Then you will perform a verification of robustness. So it means that you will modify a few parameters in a short range and check that it does not affect significantly your resolution. And if you have observed some problems during your robustness, you can go back to the optimization to optimize the separation. And finally, you will have all the documentation that will be produced automatically by DryLab that will summarize all the method development steps from the design of experiments to the robustness. So this way of showing the workflow is quite similar, but it shows also how it can be automated using Mpower. So there is an automation module within DryLab that allows to perform automatically some tasks with Mpower. So from the method target profile to the knowledge management documentation, Mpower will automatically interact with DryLab. So first of all, when you perform the design of experiments, you will use the automation module to create your batches, as I will show in the next slide. Then you will process your data within Mpower, and DryLab will recover all the data that were processed in order to perform the design space modeling. Once you have set your set point, you will send all the information to Mpower to confirm that the set point and that the modeling was correct, and that you obtain experimentally the same data that you have modeled within DryLab. And finally, for the robustness assessment, you will send all the information to Mpower in order to verify the robustness results. One thing that may be interesting also for small molecules is that for the peak tracking, DryLab can use MS data that are obtained within Mpower using a mass detector such as QDa or SQD2. So now in the next slide, I will show how the interaction between DryLab and Mpower is done. So first, you have here the DryLab data entry page on which you can see the experimental design. So the different parameters, the different variables that you will use in your design, and the different parameters that you will study. You have information about your column, about the instrument, and also information about the gradient data. So from 5 to 80% of phase B. So the examples that are shown in this slide and in the next slide are focused on a small molecule example. For proteins, it will be slightly different, but the approach is exactly the same. So once you have done all this, you have here all your runs that are created and that can be exported to Mpower to create the DryLab 3D model. Here you have the DryLab automation window. So in this window, you will set additional information about the instrument setup, the number of solvent lies, the wavelengths, and so on. You will have information and options about your method, the run-to-run equilibration. So between each run, how long do you have to equilibrate your column, and the design of experiment injection order. And finally, the complete gradient program. Once you have that, you click on Run, and everything is created by DryLab with Mpower. So you will have here all the objects that are created by DryLab. So first, the sample set, the 12 instrument methods corresponding to the 12 runs, and the 12 method sets. So here is the sample set that is created by DryLab. So for each method that you see here, from method 1 to method 12, you have the equilibration step and the acquisition step, with the number of the plate, the injection volume, the number of injections, sample name, method set, and so on, with all the runtime. At the end, you can calculate easily what will be the sample runtime, but also what will be the sample set runtime. And you see here that for this example of 12 runs, so that's usually the maximum that you have when you perform a DryLab model, you have a run of about 5 hours. Then you perform the acquisition within Mpower, you process your chromatograms, and then the chromatograms will be retrieved by Mpower. So you have here all the filtering options, the 12 processed runs that are imported back into DryLab, and additional data about the peaks. Then the next steps within DryLab is to perform the peak tracking. So if you don't use the automation module, if you don't process within Mpower and don't import back into DryLab, you will have to do the peak tracking manually to check that if you have some difference in selectivity, for example, to tell DryLab which peak corresponds to what. So some things are done automatically. Sometimes if you have some coelutions or some differences in selectivity, you will have to attribute manually the different information. If you perform the process directly within Empower, it is done automatically. And that's the way also, as I said earlier, for small molecules where you can use MS data. Once everything is processed, you have your design space modeling with the most important thing is the resolution map here. And if you have a 3D model, the design cube that is shown here. So if you are in the red zone, it corresponds to a zone where the critical resolution is good and where you have more than 1.5 resolution, for example, between your critical pairs of peaks. So you see here the chromatogram, you have here the gradient, and it's here that you can choose to modify the gradient to optimize your method. So I cannot show here because I have no video, but you can move the different dots here that are here to change the gradient, the beginning and the end of the gradient, to add another step, to do a step gradient at some times, to increase the runtime or decrease the runtime. And when you do that, you have automatically on this window the update of the chromatogram. So you see in real time what the modification in the gradient has as an impact on the chromatogram. And that's where you can optimize your separation and choose the set point somewhere here. Once you have defined the best conditions that you have, you will have to verify that experimentally you have exactly the expected chromatographic profile. So here, once again, the conditions will be sent automatically within Empower with the instrument method, the sample set, and so on. You can analyze several samples. So here you see that we have 2 samples that will be used during the verification run. And you have these results as a verification of send point. So here in green, you have the model chromatogram within DryLab. And then in blue, the chromatogram that is obtained experimentally within Empower. And you see that in this example, you have a perfect match between the model chromatogram and the experimental values. Finally, the next step is to evaluate the robustness of the set point. So if you modify your conditions, will it have an impact on the resolution? So you see here that you can modify, for example, the flow rate, the dwell volume, if you expect that you may move from one chromatography system to another, the temperature, and so on, the gradient points, and automatically DryLab will generate all the chromatograms that would be obtained within this range of conditions. And you have here on the 6 first rows, the worst case conditions for which you will have the least resolution. What you see here in this part is the fact that everything is blue. So if it's blue, it means that you reach your critical resolution, so higher than 1.5. If you have some points that do not comply with your expectations, it would be in red. So here you see that you have a robust method that within this range of conditions, you will still have your critical resolution. And here, finally, you see the impact of the different parameters on the resolution. And you see here that the most critical parameter is the starting point of the gradient, the percentage of B at the beginning of the gradient. It's something that has the most impact on the separation. But anyway, even with when in this range, so the start of B is plus minus 1%, in this range, you still have a good resolution at the end. So I've shown this example, so I will now show you how it works in practice. And I have taken examples of monoclonal antibody subunits, and the first case study is the separation of these subunits by reverse phase chromatography. So why would you want to analyze monoclonal antibodies at the subunit level? So as you probably know, monoclonal antibodies are very complex molecules of about 150 kilodaltons that have many modifications, including glycosylation, deamidation, oxidation, and so on. So analyzing those molecules at the intact level is quite challenging, because in terms of chromatographic resolution, especially in reverse phase, you won't get a very nice resolution on these antibodies. So if you do mass spectrometry also at 150 kilodaltons, the mass resolution and mass accuracy won't be perfect. So one way to circumvent this issue is to use an enzyme called IDS, or Fabricator, commercialized by Genovis. And if you use Fabricator and then you reduce the disulfide bridges, you will obtain these different species that are all about 25 kilodaltons. At 25 kilodaltons, you have an increased chromatographic resolution, but also an increased mass accuracy if you use mass spectrometry. So it's very interesting to use this approach, but as all these molecules have a molecular mass of about 25 kilodaltons, it may also be challenging to separate them by chromatography. And it's very useful to use a software such as DryLab to optimize this separation. So a few words about the retention mechanisms of proteins by reverse phase. So it's called an on-off retention mechanism, so bind and elute. If you have a look to what happens to small molecules, you see here on this example that a 3% decrease in the percentage of phase B in a separation will lead to 1.5 modification in the retention factor. If you have a look to insulin now, that is about 6 kilodaltons, a 3% decrease in phase B will lead to 15 times modification in the retention factor, and it will be even higher for larger proteins. So it's clearly very different, and you can fine-tune your separation only by modifying your gradient very slightly. So it means that for protein separations, it will be very useful to use multilinear gradients. So for example, if you start from a generic gradient where you obtain this separation between FC, LC, and FD, you can see that if you increase the initial gradient steepness, you can get rid of all this zone that is not really interesting in the separation. If you had a time point here after the first two peaks, you can then also slightly modify the retention of the third peak, and then you can here increase the steepness of the third segment in order to move the third peak in smaller retention times. And finally, you can cut all this last zone of non-eluting peaks. So you can start from a 10-minute run to a 4-minute run with a better resolution and also, of course, a gain in efficacy. So this can be done experimentally, but to do that experimentally without the use of a software, it can take a lot of time, and using DryLab for this optimization is very interesting. A few words about the temperature. So temperature for protein separation is very critical. So as you can see here, for intact rituximab or reduced rituximab, if you use low temperature at 40 degrees, for example, you detect nothing on the chromatogram for the intact mAb. For the reduced mAb, at 40 degrees, you have broad peaks and a low recovery, and you get very nice peaks and very nice separation and recovery only at high temperatures of about 80 degrees. So you will need elevated temperature to limit absorption in reverse phase chromatography of proteins. So if you have a look now on the retention property of monoclonal antibodies in reverse phase, so compared to small molecules, in terms of gradient steepness, you don't have a linear behavior, and you have rather a quadratic trend. It is not really critical if we have to use a modeling software such as DryLab, because on a narrow gradient range, you will still have a linear approximation that will be okay. So as you can see here, on a small range, you have something that is almost linear. So it will be possible to use a linear model within DryLab. Regarding the temperature, we also have an important deviation from Van 't Hoff type behavior, but for the high temperature range, you also have a trend that is close to linear. So in our case, at high temperature, as I said earlier, you will have something that is quite linear, and you will be able to use a linear model within DryLab. So we decided to apply this to the development of the separation of nivolumab subunits, and we focused also on the oxidized species of the FC fragment that we wanted to separate from the non-oxidized species. So we used a bioresolve RP column, the mobile phase that are 0.05% TFA and 0.05% formic acid in water and acetonitrile, a flow rate of 0.4 milliliter per minute, injection of 1 microliter, and a two-dimensional model, so 4 runs with 2 temperatures, 65 and 85 degrees, and 2 gradient run time, 8 and 20 minutes. So these are the results of the initial runs that we have to perform with DryLab. So we have 4 runs, 4 chromatograms that you can see here, with the 3 main peaks that we expect, and the oxidized species here in red. So it corresponds to the oxidized FC fragment. Once we have this, we are able to compute the resolution map within DryLab. So you can see here that you need something of higher runtime higher than 5 minutes to get a good resolution, and that the temperature has no significant impact here on the separation. So you can see here at this set point that we have here, we have a very nice resolution that is modeled between the different peaks and also between the FC and the oxidized species. So we can then optimize the gradient, as I said earlier, by adding some points in the gradient to remove this zone here that is not really useful, and to also optimize the separation at the end of the gradient. And once you have your set point, you can verify that everything is correct. So here, that's the set point that we have. The gradient is shown here. We have the separation between the FC and the oxidized species, LC-FD. So the upper chromatogram corresponds to the theoretical separation within DryLab. And you have here on the bottom chromatogram what we obtain with mPower. So you see that we have a very good agreement both in terms of retention times and resolutions. We only have a difference here because we separate some variants of the FD chain that were not taken into account in the model. But we have a very good agreement, and we are able to have a very good separation that was optimized quite quickly with only 4 initial runs. We also studied the robustness. So you have here the flow rate, the dwell volume, the temperature, and so on, with some tolerance that correspond to the qualification of the equipment. And you can see here that everything is blue. That means that the critical resolution that corresponds to the resolution between the FC and the oxidized FC is always higher than 1.7, whatever the conditions that you will use during your robustness study. So we were able to develop very quickly a reverse phase separation of MAB subunits. So the second case study is still focused on subunits of nivolumab, but now using HELIC instead of reverse phase. So why use HELIC? So first, it's a more recent approach that was possible for proteins thanks to the availability of wide pore HELIC columns. It's a method that is orthogonal to reverse phase LC, and the main advantage, as you can see here, this image comes from an application note from Waters. You have here what I've shown before by reverse phase, and on the bottom chromatogram, what you get on HELIC. You have the separation between FD and LC with a difference in selectivity, but you have also a separation of the different FC glycoforms, and that may be very useful if you want to study the glycosylation of your antibody. But the development of HELIC method is quite tricky, and it was very useful for us to try and optimize this method using a software such as DryLab. So in HELIC, you also have an on-off mechanism. For the separation, you see that you have a very steep linear-like trend. So for both the IDS digested and reduced cetuximab and trastuzumab shown here, so you will be able also to use multilinear gradients in order to optimize your separation. The impact of the temperature on the retention and recovery. So you can see here that you need a temperature higher than 70 degrees in order to get a good recovery. So that's an example shown on NIST-MAB. You see that at 40 degrees, you have tailing and you have a lower recovery. But what is interesting is that we have a perfect linear relationship in that range. So it means that we will be able to use a linear model within DryLab. If we have a look to the impact of the gradient steepness now on the retention, once again, we have a near perfectly linear relationship in the practical range of the separation. And once again, we will be able to use a linear model with DryLab. So at the end, we'll have our main method variables that can be the gradient time and the temperature. And due to the fact that we have linear models, we can use only 4 runs to create our resolution map. So we used a Waters glycoprotein amide column, a white pore column, with mobile phase that are 0.1% THC phase in water and acetonitrile, a starting gradient from 25 to 40% of B, a flow rate of 0.4 milliliter per minute, injection volume of 1 microliter, and this two-dimensional model with 2 temperatures, 70 and 90 degrees, and 2 gradient times, 10 and 20 minutes. So once again, we have our 4 initial runs that were performed in order to build our model. You see here the FD, LC, and the FC glycoforms that are shown here with some minor variants that are also detected. And with these 4 initial runs, we have built our resolution map. So it's a 2D resolution map with the temperature and the runtime. You see here that if we want to separate our critical pair that is shown here in red, and that corresponds to the 2 isomers of G1F on the glycosylated FC fragment, you need to have a long gradient. So before 16 minutes, you won't have enough resolution. So you need a long runtime in order to separate this. But anyway, for the other peaks, the separation will be okay. You also see that the impact of temperature is not very significant. You have a better resolution if you increase the temperature, but it's not critical. So even at lower temperature, if you are okay with increasing the runtime, it should still be okay. So once you have that, we have set our set point, and we verify with Empower that the modeling was okay. You can see here that for FD and LC, it's okay, both in terms of retention time and resolution. For the FC glycoforms, we have something that is quite different. So you see here that the peaks that we obtain experimentally show some fronting. So we think that it is due to isomers. So we have done a test to check that it was not due to the lysine variants on the heavy chain. And we have tested that with carboxypeptidase B digestion, and we have no improvement of the peak shape. So currently our thinking is that we begin to separate some isoforms of the glycans by the helic that cause this peak fronting. So work is still in progress. We are still studying that. But what is interesting anyway is that with helic, that is not really easy to develop. DryLab is able to predict the chromatogram and the behavior of the chromatogram quite easily. And we see here a very nice agreement between the theory and the experimental chromatogram. So now a few take-home messages. So software-assisted method development, so here using DryLab, is a powerful tool for efficient development of LC separations of biomolecules. So the separations of map subunits by reverse phase or helic can be optimized in a single working day with DryLab. And Empower automation module is a valuable tool to further automate the development. So you don't have to create anything manually within Empower. Everything is done automatically by DryLab. So I will finish by thanking my colleagues at Quality Assistance, so Morgan and Claire, who worked on the project. Shabox Fekete from Waters, who helped us a lot for the implementation of DryLab for this application on map subunits, and Arnold and Magdalena from Monna Institute for their help also on preparing this presentation. And finally, I thank you for your attention, and I will be happy to take any question. If you have further questions after this presentation, feel free to contact me using the email address that is shown on this slide. I thank you.
Arnaud, thank you for sharing how Quality Assistance implements DryLab as tools to support QBD method development and as a service to enhance confidence in your customers' methods. Please enter your Arnaud in the box, and we will try to address them during the live Q&A sessions.